The overall goal of this protocol is to demonstrate the development of a customizable gold nanoparticle-based immunoassay capable of analysis using UV-visible and Raman spectroscopy. The use of two optical methods provides internal assay validation and increases the assay's flexibility.
This method answers key questions concerning the development of a gold nanoparticle-based immunoassay and explains how functionalization of the nanoparticles used in this assay will influence optical absorption and light scattering.
This protocol is unique because it incorporates materials found in typical research labs to fabricate an immunoassay platform that can be customized to fit individual research needs.
The immunoassay is based on UV-Vis and Raman spectroscopy. Using Raman spectroscopy is better than fluorescence or colorimetric methods as it has narrower bandwidths, allowing for the detection of multiple biomarkers at the same time.
To begin, hydrate the bifunctional cross-linking agent orthopyridyl disulfide PEG NHS in 100 millimolar sodium bicarbonate. The orthopyridyl end will bind to the gold nanoparticle surface, and the NHS end will react with a primary amine on the protein surface once activated. Be sure to make the cross-linking solution within approximately 20 minutes of when it will be used. Next, add the bifunctional cross-linker to the antibody solution at a 2:1 conjugation ratio. Be sure the antibody solution is free from carrier proteins that may interact with the reaction. In a separate microcentrifuge tube, add the bifunctional cross-linker to the antigen solution at the same ratio to be used for the control.
The 2:1 ratio is assuming a 50% conjugation efficiency.